Okay, good morning, and we are ready to start our AP1 lecture today. We're still in chemistry. We're trying to wrap that up maybe today. Who knows? We'll see how it goes. I'm going to start again, pick up in lipids and go from there. And just a review of a couple slides before we get into move into new material. Does anybody have any questions on anything at this point? Okay. Well, if you have a question and you don't want to ask it in class, that's fine. Shoot me an email and I will get back to you right away. Okay, so I'm going to go ahead and open up the share screen. Hopefully the PowerPoints are even there, so that's good. So, okay. Let's get this going. Now, we were talking about organic compounds, and we had finished up our discussion on carbohydrates. We know carbohydrates are sugars, the sugars, the pastas, the starches, things that we get from plants, whether it's potato or whole wheat bread or sugar. You know, sugar like we get in a Coke, the high fructose sugar, which is a simple sugar related to glucose, or we have sugar added to our coffee, that's table sugar, sucrose, which is a disaccharide. You know, there are three types, mono, di, and polysaccharides. We know that our sugars that we store in our bodies, we store as a polysaccharide of glucose, long chain molecule that we end up calling glycogen. Now, if we take in too much sugar, more than our bodies can store as glycogen, we're going to convert it to a lipid and store it in the adipose tissue. And that involves, additional, retention. It may involve weight gain. A person who takes in more sugar that they can consume or store as glycogen will often experience a weight gain. Now, the lipids are a little bit different than the carbohydrates. Still organic molecule, still contain carbon, hydrogen, and oxygen. It's not always a 1 to 2 to 1 ratio like it was in sugars. Sometimes we throw phosphorus in there. Sometimes we have some unusual ring structures in the lipids. So we've got the triglycerides, those are the neutral fats. That's the stuff that we eat. That's the stuff that we store in our adipose cells. We have the phospholipids, which are the main component of our cell membrane. And we have steroids, which are used to make hormones. Hormones serve as chemical messengers in our bodies. And these chemical messengers can be like the sex hormones, for example, testosterone and estrogen. These are steroids. They're all based off cholesterol. Of course, cholesterol gets a bad reputation, but it really isn't a bad molecule. It's just, if it's present in our bodies in large quantities, does it cause health issues? However, cholesterol is very important. Cholesterol is the #1 steroid. All other steroids are derived from the structure of cholesterol, and cholesterol, builds reinforcing material for inside the cell membranes. We derive all of our hormones off of cholesterol. And if we didn't have cholesterol in our diet, we'd still make it in our bodies. And the eicosanoids are a type of molecule that we release to help deal with inflammation. We'll talk about that when we get into that. So the triglycerides, the neutral fats, They are a fat, and a triglyceride is called a fat when it is solid at room temperature. Things like butter, things like animal fat, lard. The shortening lard is a fat. It is a solid triglyceride at room temperature. There are oils when they're liquid at room temperature, things like corn oil and olive oil, canola oil, sunflower oil. All these vegetable oils are liquids at room temperature, and they're all made the same way. They have 3 chains of fatty acids, which are long molecules of carbons and hydrogens and oxygens, about 20 to 30 carbons long. There are 3 of these chains that are bonded, covalently bonded to a 3-carbon glycerol. Glycerol is an alcohol. Whenever you see the ending OL on one of these words, it means it's an alcohol. Glycerol is an alcohol version of glycerin. Ethanol is the alcohol version of ethane. You know, ethanol is the stuff that shows up in beverages. It's stuff that shows up in fuel additives, you know, ethyl alcohol, the stuff that when we have corn alcohol added to gasoline, that's ethanol. So now the purpose of all these fats is primarily energy storage, because a gram of fat has much more available energy than a gram of carbohydrates. A gram of carbohydrates contains 4 calories of energy. 1 gram contains 4 calories of energy. A gram of fat contains 10 calories of available energy. So the calorie makeup that we read on a food label, based on the presence of carbohydrates and fats and proteins, is the sum of all the available calories in there. So if you have 10 grams of carbohydrates, that's going to represent 40 calories. If you have 10 grams of fat, that's going to represent 100 calories in there. Proteins don't contribute a lot to the calorie makeup. So, but they you know, usually because protein, most food substances like fruit bars and cereal and apples, the amount of protein is pretty low in plant-based foods. So anyway, most of what we see from fats and lipids, as well as the carbohydrates, are storing energy. We store some of it as a carbohydrate, as the polysaccharide glycogen, and the rest we can store as adipose. We use fats for, and when I say fat, I mean a lipid. We use fats for insulation. We have a layer of fat underneath the hypodermis, or in part of the hypodermis in our skin. You know, we have the epidermis, the outer layer, the dermis, the middle layer. We have the hypodermis below that, and that's where we have the adipose cells, and that's where we protect ourselves by insulating from heat and cold. And we have protection. A lot of our organs have no protection around them except a layer of fat. Our kidneys, set up very high, right below our diaphragm, embedded in our back wall of the abdominal cavity, they have no protection. They have no bony structures around them like the heart and lungs do, or like the brain does. So the kidneys are going to have to embed themselves in a layer of fat for protection. Now, your typical triglyceride looks like this. This is glycerol out here on the far left. Glycerol is a 3-carbon alcohol. It is the glycerol molecule has 3 carbons and 3 alcohol groups attached. Those little OH groups are alcohol groups. You don't need to remember any of that. Or you know, this is not a class in organic chemistry, but glycerol has 3 carbons. And the fatty acid chains about 20 to 30 carbon atoms long, with lots of hydrogen attached, and what we call an acid group here at the end. Again, this is not organic chemistry, but the acid group has a carbon, a double bonded hydrogen, and a hydroxyl. It's not an alcohol. We call it an acid. And so that function, we call it a functional group. That functional group on the end of it makes it an acid. Now, what do you have to remember? That's a fatty acid. And a fatty acid chain always has an acid functional group on it. Fatty acid chains are about 20 to 30 carbons long, and they attach to the glycerol, and they give us the triglyceride. There are 3 fatty acid chains attached to the glycerol. And so we see that this is what a triglyceride, a neutral fat, looks like. You may have heard the term triglyceride, you know, when you had blood work done, for example, and you want to get your triglyceride levels. What is the you know, the fat level in your blood, you know, circulating in your plasma? And so we see, you know, we measure the triglyceride levels. So this is what a fat looks like. So 3 carbon alcohol with 3 fatty acid chains attached here. And you can see that there's 2 hydrogens for every carbon all the way out to the very end group. The end carbon has 3. So every, you know, in some of these fatty acid chains have every available space for a hydrogen filled up. If you have every space filled up, you are saturated. We call it a saturated fatty acid. All the covalent bonds in that chain are going to be single covalent bonds. So a carbon atom linked is a carbon atom is linked to 2 other carbon atoms and is linked to 2 hydrogens. I can show you that here. There's a carbon right there. Well, we use this one. Now we use this one right here. That carbon is linked to that carbon. That carbon is linked to that carbon. This has a hydrogen here and a hydrogen here. This has a hydrogen above and below, and this has 3 hydrogens because it's the end group. Every space available to bond is filled up with single covalent bonds. And every space that can hold a hydrogen has a hydrogen. So we have the maximum number of hydrogens available here. We say it's saturated with hydrogens. And these are the animal fats. These are the animal fats. Saturated fatty acids show up in the animal fats, which are solid at room temperature. Butter is an animal fat. And because it comes from cows, from milk. So it's an animal fat. Butter will be solid at room temperature because it has all of its bonds held together with single covalent bonds. It has the maximum number of hydrogen atoms available. It's saturated with hydrogens. Butter, lard, the solid fat that you have on a piece of meat that you trim away, those are all going to be saturated fats. If there is an unsaturated fat that occurs, then it's because there's a double bond somewhere. See, if you look at the saturated chain here, this is a better picture I'm showing you. Up at the top, all the carbons are linked together with single covalent bonds. And that still leaves room for 2 hydrogens, one here and one here on every one of these. So every space is filled up with a hydrogen atom. But what happens if we were to have a double bond between the carbons? Instead of sharing 2 electrons, we're sharing 4 electrons. This double bond here prevents another hydrogen from attaching here and here. So we don't have full saturation because we don't have the electrons to share. those electrons are shared in the double bond there. When we have a double bond, something unique happens to that fatty acid chain. It twists, it kinks, it bends. It bends at the double bond. And so this is, we call this an unsaturated fatty acid. Perfectly normal, but this is an unsaturated fatty acid. And the presence of the double bond will determine whether the fat is a liquid or a solid at room temperature. Now we know that if it's a saturated fat, it's always going to be solid at room temperature. We know, now we see what an unsaturated fat looks like. Unsaturated fats are liquid at room temperature. So we say that the fatty acid chain is saturated if it has no double bonds and it has the maximum number of hydrogen atoms attached to the carbons. An unsaturated fatty acid has a double bond and causes that chain to kink. We call it polyunsaturated if the fatty acid has two or more double bonds in here. So they're either saturated or unsaturated. And if you're unsaturated, you can have one double bond, or if you have if you have two or more, you're polyunsaturated. These saturated fatty acids are solid at room temperature thanks to van der Waals attraction. Remember van der Waals? Van der Waals occurs when you have chains of molecules that are close together, and the random movement of the electrons in the covalent bonds can create slight positive and negative attractions to those chains of molecules. So remember what van der Waals is? Yeah. Van der Waals, it's like the fourth type of bonding. It's like 1/100th the strength of a hydrogen bond. What happens in van der Waals is electrons are spinning in their orbits around the nucleus of all the atoms, and every now and then some more electrons end up on one side than the other side of the orbit. And so for a millionth of a nanosecond, that side of the atom of the molecule becomes slightly positive or slightly negative, and it's attracted to another molecule which is in very close proximity to it, and they bond together and then they let go. They bond together and they let go. It's a magnetic type of attraction. Van der Waals attraction is what occurs in these fatty acid chains. These saturated fatty acids are lined up. Let me go back here. Right here, these are lined up in close proximity to one another. And as they're saturated, they're going to be attracted to each other through van der Waals attraction, which is like a magnetic attraction. It is very brief. It's transitory. It occurs randomly, but it does occur. And when these fatty acid chains have van der Waals attraction going on, which they do, they're going to be pulled together and they're going to become a solid. Now, at any given time, any number of these are attracting and unattracting and attracting and unattracting, or letting go or whatever. But the fat stays solid at room temperature. Animal fat, butter, lard, the actual fat itself from a meat product is in there. And that's, and so they're solid thanks to van der Waals attraction. They have to be saturated and they have to have van der Waals going on. If they're saturated, they're likely going to have van der Waals attraction going on. So unsaturated fats don't have van der Waals because the unsaturated fat has at least one double bond in its fatty acid chain. And the molecule kinks. The fatty acid molecule kinks at the double bond and it pivots. And so it's no longer attracted to the chain next to it. It doesn't have the random attraction like van der Waals occurs. Van der Waals doesn't occur when you have an unsaturated fatty acid or a polyunsaturated fatty acid. Since there's a bend in the molecule and it's pivoting at that bend, it never gets close enough to the other fatty acid chains for van der Waals to work. And so all of our plant oils are going to be, all of our plant fats are oils. And they're going to be liquid at room temperature. Olive oil, corn oil, sunflower, safflower, just plain old generic vegetable oil, which is a mixture of oils, canola oil, all these things that you see, at Food City or at Kroger's, wherever you shop, have all these oils in there. They're all pretty much the same. They're all going to be pretty much the same. They're going to be low in saturated fats and high in unsaturated fats because of these double bonds here in their chains. Plant fats are oils because they all have double bonds in there. Now, the unsaturated fatty acids tend to be better for us in our diet. The unsaturated fatty acids with that double bond in there, all the plant oils, olive oil, as I said, that's a good example. The unsaturated fatty acids contain what we call omega-3 fatty acids. These are the so-called heart-healthy fatty acids. The saturated fats can form something called trans fats, and oils that have been modified, because oils can be changed, and they will form what are called trans fats. And these are unhealthy fats for us. So we find, though, that all in all, the unsaturated fatty acids are healthier for us, particularly in terms of our blood circulation and our heart, than saturated fats. Does that mean you can never eat saturated fats? No, of course not. It means you eat those in moderation. You eat more unsaturated fats in your diet than saturated fats. There's nothing wrong with saturated fats as long as that you eat them in moderation. There's nothing wrong with just about practically any food. The magic term here is always moderation. So here, this might, if it works, it should show you this.
Butter, lard, margarine, and salad oil are composed of lipids called fats. To make fat
Okay, so what you're seeing here is this is a saturated fat right here, and the van der Waals attraction is occurring across here. But if we had a double bond in any one of these chains, that would make this fat a unsaturated fat, and it would be liquid at room temperature. Common unsaturated fats, we have butyric acid. Okay. There's a double bond. I'm sorry, that's oleic acid. This is butyric acid. It's the double bond's only on the oxygen here. So that's a saturated fat. Every space available. It's a little fat. It's only 3 carbons long. I was looking at the functional group. This is for the functional group for the acid. 3 carbons long. It's a little fatty acid chain, but it is still a saturated fat. But here we see a longer chain of oleic acid, and you don't need to remember what they look like. Okay. If it has a double bond in there, it's an unsaturated fat. This is what we call a monounsaturated fatty acid, 1 double bond. Here is linoleic fatty acid, It is polyunsaturated because it has two double bonds. It'll have a double kink. It will not be attracted with van der Waals to any other fatty acid chain. Problem that we encounter with unsaturated fats is we can change them. We have the ability to change unsaturated fats, and through the process of hydrogenation, we can break the double bond and force hydrogens onto the unsaturated fat. This happens all the time in the food industry. Vegetable oil gets hydrogenated. Corn oil gets hydrogenated to form margarine. Margarine is it is not a natural product. Margarine is a chemical product. You saturate the oil, saturate corn oil, it becomes you break the double bond, that corn oil becomes solid at room temperature. Now, margarine, of course, is the butter alternative. Everybody knows what margarine is. It's promise, it's heart healthy, it's shed spread, it's country crock, it's any of this stuff, and you use it in place of butter. The only thing about it is you have to add flavorings, and you have to add coloring to it. You have to add salt to it to give it a tasty flavor, because it doesn't taste quite like plain ordinary margarine, just hydrogenated, isn't going to taste like anything. It's going to be a white, bulky mass. And so before it can be sold, it has to be colorized with some sort of buttery looking color. It has to have a buttery flavor added to it. It has to have a little salt added to it to give it a little better flavor, because it's pretty bland even when you add the buttery flavor to it. Probably, 80 years ago or more, margarine, the dairy industry wouldn't allow margarine to be sold if it was flavored or colored. People would have to buy, because they wanted to have margarine. They could buy the margarine at the store, but it would come with a flavor pellet, a little piece like a pellet that you broke apart and mixed it, and a dye pellet, and you mix this all together, and you'd have your margarine supposedly looked and tasted like butter. I think it was very common during World War I, maybe World War II. Maybe it's 100 years ago. I don't know. The dairy industry is very specific about this. However, now you go into the dairy counter of the dairy section in Walmart or Food City, and you see lots and lots of space available for margarines, and a little bit less space available for butter. Butter has taken a very bad hit over the years as being evil, harmful. Again, how much butter or margarine do you eat on a daily or a weekly basis? Just keep in mind that there are a lot of additives in margarine to make it taste better for us. So it all comes down to, pick your poison. It all comes down to moderation. Do you eat butter or do you eat margarine? So now, one of the problems with hydrogenation is it can create something known as trans fats. We have cis fats and trans fats. It has to do with which way the molecule bends. Does it bend to the left or does it bend to the right? One of them is a trans molecule, and one of them is a cis molecule. Trans fats, it has to do with direction, the shape of the molecule. A trans fat is going to be solid at lower temperatures than the cis fats. And so these trans fats have been considered a good thing in the food industry because it keeps foods stable and has a longer shelf life than than the cis fats do. You know, you can go buy, you can put a tub of margarine in the dairy section and leave it there for a year, you know, or whatever the expiration date is. These are, it holds the flavor, it holds its texture. It can stay in the shelf much, much longer. And that was the advantage of trans fats. Now you'll notice if you ever read a food label that trans fat contents are now 0, because trans fats are not, you know, a healthy fat. Just because they are more stable at room temperature and they are solid at room temperature, they mimic the animal fat. There we go. This is you know what I was talking about over here. This is how margarine had to be processed at home with a flavor pellet and a dye pellet, or as I say, the color berry in here to mix it up. And over here, of course, this is how we buy it now with Country Crock, and I can't believe it's not butter, and just plain old generic margarine, for example. You know, you see it looks like butter. It sort of usually tastes like butter. If you haven't had real butter, you know, you don't really notice the difference in here. Margarine is, you know, the oils start out as unsaturated, but then they're broken down by hydrogenation. They are modified by hydrogenation. They become solid. Think of, what about, you know, consider Crisco. Crisco shortening, you know what Crisco is. It comes in the blue can. You use it in baking. Crisco is corn oil that's been hydrogenated, and it solidifies. And it's used in lieu of lard. Lard was the original animal shortening. Shortening is put into pastries and pie crusts and biscuits. When the purpose of the shortening is to make the biscuit or the pie crust light. As the shortening that's added to the dough breaks down under heat, it releases carbon dioxide, and that causes the dough to flake into layers, and very lightweight layers. You know, and so the pie crust is a lightweight pie crust. The biscuit becomes light and fluffy. You know, if you don't have shortening, you know, you're trying to eat a hockey puck or something like that. You know, a biscuit without shortening is dangerous because it's very heavy and dense and can break your teeth. Not really, but you know, it's not a tasty product. That's why we use shortening. The best bakers still use lard as their shortening, because nothing works better than lard to make a light and flaky pie crust or pastry shell or biscuit. But again, how often, you know, do you eat biscuits? How often do you eat pie crust? How often do you eat any kind of pastry shell? You know, is it, you know, or does it matter? So, okay, what about trans fats? Trans fats show up naturally. You know, milk products, meat products, milk itself all contain trans fats. They and they're allowed. You know, you can't take them out of those kind of animal type foods. You know. However, the trans fats that are formed when fatty acids are hydrogenated are now banned. So you know, they're not allowed. You know, they have to be hydrogenated without the fatty acids. So why do we care about all that chemistry? Yeah. Because trans fats increase the level of the bad cholesterol, what we call the bad cholesterol in our blood. It's what we call the low density lipoprotein cholesterol. Low density lipoprotein. You know, you have two types of LDLs. There's low density and high density. Low density is the bad cholesterol. Low density cholesterol takes fats and cholesterol, transports it from our liver out to the cells where it accumulates, particularly in blood vessels and around cardiac muscle, and starts narrowing the coronary arteries, narrowing the circulation in our brain. High density lipoprotein, the HDL cholesterol, takes cholesterol away from the cells to the liver for processing and destruction and removal. So if we have if we have trans fats in our diet, it increases the amount of LDLs. And so we start seeing a narrowing of the arteries in our heart and our brain and throughout our body. We develop atherosclerosis, which is the common term is hardening of the arteries. And so your patient is at a greater risk for a stroke or a heart attack. So, and that's why we pay attention to the LDLs and the HDLs. You've heard the phrase good cholesterol and bad cholesterol. Well, that's what we're talking about. We're talking about high density lipoproteins and low density lipoproteins. The high density lipoproteins carry cholesterol back to the liver, where we can break down excess cholesterol. We eat something that you think that's, let's say that you have lots of, your favorite breakfast is bacon and eggs and whatever. High in fat, high in cholesterol, higher in fat and higher in cholesterol. You have that every day. You keep adding cholesterol to your diet. Now, as long as you have the high density lipoproteins in sufficient quantity, the excess cholesterol will be removed by dragging that cholesterol that's found in the egg yolk to the liver, which breaks it down and sends it out of the body. If you have lots of trans fats available, that extra cholesterol will be taken from the liver and sent to the tissues. And that's why we pay close attention to this. Certain types of foods are considered heart healthy. Cheerios, you remember seeing the ads a few years ago for Cheerios, where it continues to advertise itself as a heart healthy diet. And it is. Any cereal product based, it isn't just Cheerios, any cereal product, excuse me, based on oat bran, like Cheerios or generic Cheerios, or oatmeal of any kind. Unfortunately, not oatmeal raisin cookies, but any cereal with oat bran will increase the levels of the high density lipoproteins and lower blood cholesterol. And so we see that when they say it's a heart healthy cereal, it's true. But then any oat-based cereal will do that. Oat bran, the outer covering of the oat kernel. Remember we talked about wheat bran, whole wheat bread versus white bread. The bran is the outer covering of the grain kernel, whether it's an oat or whether it's wheat. Now in oats, the bran, the covering here, is also indigestible. It is made of cellulose, but it stimulates the production of HDLs. So any oat cereal, any oat grain product with oat bran in it is going to be healthier for us. It's going to help, reduce our cholesterol levels. So it is heart healthy. That's a true statement. It just isn't confined to Cheerios, which is okay. But, if you like Cheerios, and most people do like Cheerios. So anyway, so a little factoid there off to the side. It is a true statement, what they say. What happens to excess fat in our bodies? As we say, excess nutrients. A person who maintains high levels of nutrient intake, they overeat consistently. What will happen is when our bodies take in food, carbohydrates, lipids, and proteins, the carbohydrates are going to, the the higher level, as carbohydrate levels in our blood go up, it's going to trigger the release of insulin. Insulin causes these cells to make channels to allow the facilitated diffusion of glucose across the cell membrane. Glucose is a big molecule. It can't get through the cell membrane without help. Sucrose, table sugar, it can't get through at all, but glucose can get through if the insulin tells the cell to open up a channel. And that's what it does. So we bring the glucose in and the cell gets fed. And if we have any excess glucose that we bring in, it goes into, it becomes glycogen. It can form glycogen in muscle cells. And if there's more glucose, it'll go into the liver and become stored there as glycogen. Glycogen is how we store it. If there is excess glucose, it is converted to fat and it goes into the adipose tissue. It's the process of lipogenesis, the creation of fats. Genesis is creation. Lipogenesis, the creation of fats. And we form these fats in our adipose cells, and the adipose tissue enlarges as more and more fat is deposited there. So, if you take in more calories than you break down on a daily basis, there's going to be weight gain. That's the whole ticket, the whole secret to weight gain and weight loss. If you take in more calories than you burn, you're going to gain weight. If you burn more calories than you take in, you're going to have weight loss. I had a friend years ago that worked for the State Department, and she was posted to Paris, really nice posting for her. And she worked at the U.S. Embassy in Paris. And in Paris, believe it or not, there's lots of places to buy pastries and breads. Who would have thought? And so it became a habit for everybody in the office when in her particular office, that when they would have their break for coffee in the morning, they would all go around the corner away from the embassy to this little bakery and have bread and butter and coffee. It sounds like a harmless snack, right? Well, she did this every day. And at the end of a year, she had gained 40 pounds, which is pretty remarkable. And it's like, well, how? Well, all the excess carbohydrates from all that bread, all those different types of breads and rolls and pastries were more than her body was accustomed to taking in. And since she was in a rather sedentary position where she didn't get a chance to move around a lot, she was consuming more calories than she was burning. And so at the end of a year, she had a weight gain of 40 pounds. Lipogenesis, it was not her friend. Now, when we need that energy, so how did she get it off? Well, she exercised and she worked out and she watched her diet. And so she forced the stored fats in the adipose tissue. It isn't the adipose tissue itself, it's what the adipose tissue stores. Adipose tissue will break down the fatty acids through when there are energy demands, increased exercise, less calorie intake, and so on. Lipolysis, the lysis of the lipids. Lysis means rupturing. So just like Genesis means creation, lysis means rupturing. Lipolysis is the rupturing and the destruction of the fat cells. And so the way the guaranteed absolute #1 way to lose weight is to reduce calorie intake and force stored energy reserves to be burned. And so you lose weight. You burn up. If you're burning up more calories than you're taking in, you lose weight. It's a very simple, it's a simple mathematical process. The average person burns about 2,000 calories a day. for maintenance work and activity, and we take in about 2,000 calories a day. If you reduce the calorie intake by 100 calories over the course of a month, you do it every day, you'll lose a pound. If you increase it by 100 calories, you'll gain a pound. And so, despite the short-term fluctuations. So, lipogenesis, the creation of fat from excess nutrients. Lipolysis, the rupturing of fat, the breakdown of fats from less nutrients. Okay, so those are the triglycerides. Now, the phospholipids make up our cell membranes. Cell membranes, the structure that surrounds the cell. When I showed you those slides of the simple squamous, the cheek cells, the stratified squamous cheek cells in AP1 lab, you could see the cell, you could see the nucleus, you could see where the cytoplasm was located, and you could see the edge of the cell. Well, we couldn't see the membrane, but we could see where the membrane was located. Every cell membrane in our bodies is made up of what we call a phospholipid bilayer. 2 layers of phospholipids with their heads pointed out and their tails pointed in. Phospholipids are similar to the triglycerides. It only has 2 fatty acid chains. The third, in place of the third chain, is phosphorus. And so we have the chain looks, the molecule looks something like this. We have we have the phosphate and the glycerol here, and the 2 tails here. So we have a head and tail region. When we form the bilayer, as what we call the cell membrane, the tails point inward and the heads are out. So the inside of the cell has the heads facing it. The outside of the cell has the heads facing it. And this is what makes up our cell membrane. This is what restricts the movement of molecules across the cell membrane, either from the outside in or the inside out. So phospholipids, they are a modified triglyceride. Third category, the steroids. Steroids are made up of a 4-ring structure that are linked together, and they're all derived off of cholesterol. These ring structures, they look like, that cholesterol, of course, that's what we see here in the picture, that's cholesterol. Vitamin D is also a steroid derived from cholesterol. These steroid hormones, estrogen and testosterone, as well as the hormones that come from the adrenal glands. Cortisol, you may have heard of cortisol, that's a glucose regulating hormone. We have aldosterone coming from the adrenal cortex. It helps to regulate blood pressure in our bodies. So these are all going to be the similar. They're going to have very similar structures. They're all going to have these interlocking rings. It's just the functional groups that are going to be different. We see cholesterol embedded in our cell membranes to like reinforcing rods. So cholesterol plays a very important role there. Cholesterol, when it's transported to the cells, is used for cell membrane structure. It's used to help produce various steroid-based hormones. So cholesterol into and on itself is also not evil. We still need a certain level of cholesterol in our cells. Now, if we go cholesterol free, if we were to say, I'm never going to eat cholesterol again, that would be unwise, because our bodies still need cholesterol. We can manufacture some cholesterol, but we still need to have a certain amount in our diet for the things that we need it for, like the steroid hormones, the cell structure. Vitamin D, gotta have vitamin D. We need vitamin D is how we absorb calcium in our gut. Vitamin D builds strong bones. Well, that's the old saying goes, and it's true. We get vitamin D from sunlight, and when it hits our skin, believe it or not, we make vitamin D in our skin from the exposure to ultraviolet radiation. And the vitamin D is made off of cholesterol. So for your patient to overreact on cholesterol and say, I'll never eat cholesterol again, is a completely unwise statement. Okay, the other, last of the lipids, the eicosanoids. The eicosanoids are made from fatty acids, and they sort of get characterized as hormones. We'll talk about them again in AP2. The eicosanoids contain one of the compounds in eicosanoids are called the prostaglandins. And we use these to control, among other things, the inflammatory process. The inflammatory process is how our body reacts to injury and trauma. If you have a cut or a scratch on your skin and there's a red area around that cut or scratch, that's the inflammatory process. It is how we heal. The redness is there not because of a bad reason. The redness is there because it's increased blood flow. Increased blood flow brings more oxygen, brings more nutrients, takes away dead and dying cells and any bacteria that's present. That's what the inflammatory process does. Prostaglandins help to control that. Prostaglandins help with blood clotting and blood pressure, and even in labor contractions. To a lesser, all to a lesser extent here, but the prostaglandins are probably the best known of the eicosanoids. So, and that's the other category of lipids. So that leads us into proteins. About a third of our cell mass is protein. We use them for structure and we use them for function. They can be a very strong structural molecule. They can be a functional molecule that's very squishy. So proteins can either be fibrous or globular. The fibrous proteins are what we have in our muscles, and the collagen fibers that hold our muscles to the bones, very, very strong protein molecules. The globular proteins are the enzymes that control chemical reactions and the hormones. And most of our hormones in our bodies, like growth hormone, for example, or prolactin, stimulating milk production, are based off of proteins. Most of our, epinephrine, a hormone, is based off of protein. So they're globular, they're squishy, but they function as chemical messengers. So how's a protein made? Well, it contains carbon and hydrogen and oxygen, just like the carbohydrates and the lipids, but it also has nitrogen, and occasionally we'll have phosphorus in here too. So the proteins are made up of something called amino acids. Amino acids are the monomers that make up the polymer chain, if you will. And the key phrase here is they are held together by what we call peptide bonds. Peptide bonds. There are only 20 amino acids. And the peptide bonds are just ordinary covalent bonds. But even though there's only 20 amino acids, we can make a protein of any length by repeating the use of the same amino acid over and over again. The structure of the protein is dictated. The structure of the protein is dictated by the order of the amino acids. The order of the amino acids will dictate what the protein is and how big it is. So the amino acids Doesn't matter, there's only 20 amino acids. We can make any protein of any length we want. Consider this, there are 26 letters in the alphabet. And yet, you know, the last time I looked, there were like 300,000 different words in a dictionary. So out of just those 26 letters, we can make any 300,000 words. Well, with 20 amino acids, we can make every protein we need. The amino acid has a typical structure. It's always based off of what we call an amine group, and each amino acid has a different functional group that determines what the amino acid is. So this is your typical amino acid. Here's the acid portion out here. Every amino acid is going to have this right here. Every amino acid is going to have some sort of side chain to it that dictates what it does. And over here is the ammonia group, NH. That's the amine group. The amino group, the carboxyl group, never changes. Every amino acid, all 20 of them, will all have this, the carboxyl group that makes it the acid, and all have the amine group here. That makes it the amino acid. It is this one in the middle here with the side chain labeled R that dictates what the acid is. So, and these amino acids are held together, linked together by peptide bonds. So we start pulling amino acids together. Here's one here. There's an amino acid, and there's another amino acid. There's a peptide bond right in the middle. There's your ammonia. There's your other ammonia right there. There's your functional group there and the functional group there. And that dictates what that particular chain is going to do. The presence of the individual functional groups, the R is what we call the functional group. That's what determines what the amino acid can do. So if you're a protein, you occur in one of four shapes. Your first shape is the primary shape. That is what the protein looks like as it's being manufactured in the cell. As it's coming out of production inside the cytoplasm of the cell, we have just the basic sequence of amino acid 1 and 2 and 3 and 4 and 5 and 6 and 7 coming out in a nice straight line. That linear sequence, a nice straight line of these amino acids linked together. But they don't stay that way. They start twisting and bending right away. The linear structure is attracted to itself through hydrogen bonding, maybe little van der Waals in here will occur. And so the protein chain starts to bend and twist, and we go into what's known as the secondary structure. Because no protein stays in its primary shape. It is twisted around to assume its working shape. Now, in the secondary structure, we can have two versions. We can either be an alpha helix, where the protein coils up like a spring, sort of resembles DNA, but it's not related to DNA, but it sort of resembles it because it forms like a helix. DNA is the double helix. And this forms a shape similar to that. It's shaped like a spring. It coils up thanks to hydrogen bonds that occur across the different layers of the molecule, and it bends and twists like that. Or a beta pleated sheet, which looks like an accordion, where you have many folds in here. The tertiary structure is when you have multiple secondaries occurring along the length of the protein. Tertiary structure has lots of bends and twists in it, and it can have both the alpha helix in part of its structure and the pleated sheet version in other parts of its structure. The quaternary is a very large structure with 4 parts to it. You could say, well, it's a couple of tertiaries put together. And that's sort of true. Very large proteins are held together in a quaternary structure. Classic example for quaternary structure, hemoglobin. Hemoglobin has 4 parts to its molecule. Hemoglobin is the respiratory pigment that carries oxygen, has an iron atom based in the middle of it, and it'll hold 4 oxygen molecules, 1 in each portion of its quaternary structure. So here's what it looks like. There's our straight chain at the top, the little purple balls linked together is our primary structure. The alpha helix is shaped like a spring. The pleated sheet, the example is like an accordion. It's like a series of folds overlapping each other. The tertiary structure here, which can consist of both types of secondary structures, and then the quaternary structure here. Now, the advantage of this is that proteins function frequently based on their shape. In many places, the protein depends on its structure to cause to do something. For example, enzymes are proteins, and enzymes are very specific for what molecules they're going to bring together. And so the shape of the enzyme dictates what kind of reactants it can bring together to have a reaction occur. Or the proteins that are hormones will land on what we call their target cell. And the only way that they can work is if they have, it's almost like they're a key that fits into a special lock. And so the shape of the protein dictates where it's going to fit on its target cell. And if you're a protein, if you're a hormone, you're going to land on every cell, but you're only going to work on the ones that have a receptive site for you. And so the shape of the protein dictates what it can do. And that's why we have this secondary, tertiary, and quaternary structure. Okay. And this may summarize it even better than what I just said. Oops, let me take it away here. So this is what you're seeing here is the primary structure, just a chain. These amino acid monomers linked together with peptide bonds, and they're all held together here. And this is a straight chain. Now, what will happen is that the chain will start to twist and bend as hydrogen bonding occurs. It's already starting to, you can see here, it's already starting to twist around. Hydrogen bonding is starting to occur in places where they're coming together, and that's going to twist them into their specific and unique shape. It's going to give you the pleated sheet. It's going to give you the alpha helix shape. So now, why are those important? Well, the alpha helix is going to be present where you want to have elasticity. The protein in our hair is flexible. It will rebound to its shape. We can brush our hair back into position. The elasticity of our skin is has lots of proteins in there in alpha helix, because our skin is elastic. So we have great strength in there and the ability to rebound. The pleated sheet arrangement shows up in collagen. The pleated sheet has the proteins lined up row after row after row, side by side, giving you great strength with the collagen fibers. Consider that collagen is the most abundant protein in the body. And we will come back to this again and again. Collagen fibers are stronger than steel, strand for strand. They attach to muscles, and they attach the muscles to the bones. And collagen fibers, and they form the tendon. These tendons made up of collagen will snap bone before they break themselves. So a collagen, a tendon usually doesn't break. Of course, we have the exception with the Achilles tendon, but we'll talk about that later. That's sort of the exception to that rule. Most tendons will snap bone before they break themselves. Okay, now, let's take a look at the two distinctions. A fibrous protein, like I just been describing, they are made up of strands of protein fibers. They may be tertiary in shape, and if they're tertiary, they're going to have lots of pleated sheet, beta pleated sheets embedded in there. They're very, very stable. We find them in our keratin, the waterproofing protein in our skin, elastin, the elastic protein in our skin. Going to have lots of alpha helix in there too, because tertiary structures are made up of both alpha helix and the beta pleated sheet. Collagen, and how many times can I say the single most abundant protein in the body, as well as the proteins actin and myosin. Actin and myosin are the contractile proteins we use in skeletal muscle. These are very, very stable. They're very strong. They're very stable. The globular proteins, on the other hand, are the functional proteins. They do things. They form enzymes. They form enzymes. They form hormones like epinephrine and norepinephrine, growth hormone, thyroid stimulating hormone, prolactin and milk producing hormone. All of these hormones that are, in fact, most of our hormones in our bodies are protein based. They are squishy. They are water soluble, which creates a problem getting into the cell. Again, they are a tertiary or quaternary structure because their shape is very important. Enzymes are very critical in our bodies. Enzymes allow chemical reactions to occur at body temperature with low concentrations. The enzymes are simply a globular protein twisted around to create reactive sites on that protein for specific molecules. So you see here the example when it stops doing its thing here. Now, here we have a glucose. Well, we have a starch molecule. Lots of glucoses linked together. Now, an enzyme named amylase starts breaking down starch in our mouth. Our saliva contains amylase. It's an enzyme. The enzyme has a very specific structure just for starch. And when we expose starches that we've eaten to amylase, it starts breaking it apart into maltose, the double sugar, the malt sugar. And that malt sugar that can be further processed later on. We process that later in our digestive system. We start the process of digestion in our mouth with an enzyme known as amylase. And it will start breaking down starches into the double sugars at least. And amylase only fits on starch, doesn't fit on anything else, because the shape of the amylase is dictated by its by how it is twisted around and the presence of the alpha helix and the pleated sheet. Problems with proteins, they can become denatured. A protein is very sensitive, a globular protein is very sensitive to temperature, and it will change its shape drastically. Fibrous proteins don't worry about that so much. Fibrous proteins are very stable, but globular proteins can become denatured and deactivated. If the temperature goes up or down, if the pH goes up or down, we reach a point where the enzyme becomes deactivated and can't recover. We can operate within a narrow range of temperature rise or temperature drop or pH rise or pH drop. But when we get to extreme levels, it's an irreversible reaction. Consider this, an egg, a fried egg. The yolk of the egg is cholesterol. The white of the egg is made of albumin, which is a globular protein. If we cook the egg, the temperature change, the temperature rise causes the globular albumin to become denatured, and it cooks up into the white of the egg. You can't reverse that. It can't go back. Once it's cooked, it's become denatured and it's done. Next time you have a fried egg, you can say, hey, I'm eating denatured protein, if you feel so inclined. You can't change its shape. In our bodies, when a person becomes overheated, significantly overheated, 104, 105, 106, 107, moving up into the higher ranges of temperature, we start seeing enzymes becoming denatured and being unable to function. The same thing applies when our body temperature drops. If our pH changes, if we get too acidic or too basic, we start seeing enzymes becoming denatured. Now, if we recover from the rise or fall of pH or temperature, then the globular proteins can recover from that. But if we get to an extreme, they can't. So a person who has experienced a cardiac arrest, their blood is going to become more and more acidic because of an increase of carbon dioxide. Cells don't die when the heart stops. Cells don't die immediately. Some cells will stay alive for up to 10 or 12 hours, muscle cells in particular. And they're still busy churning out carbon dioxide from the breakdown of glucose. As long as there's available oxygen stored in the blood, they're going to make carbon dioxide. And as that carbon dioxide hits the cells, and since we're not pumping that, hits the blood, we're not pumping that blood to the lungs anymore. The heart has stopped. The carbon dioxide accumulates, making more and more carbonic acid. The blood becomes more and more acidic. Enzymes that are normally doing their job are breaking down and becoming denatured simply because the heart has stopped. The lungs aren't working. So anyway, we'll talk a lot more about that. AP2. Okay. Enzyme activity is very, very important. The globular proteins function as a biological catalyst. Remember, a catalyst is a compound that makes a chemical reaction go faster. And in our bodies, we can't do anything about chemical reactions because we can't heat the body up to higher temperatures, we can't increase the concentration, and we can't do a whole lot with the particle size. We're sort of stuck with what we've got. But we can use enzymes. We use these enzymes to allow reactions to go at a faster rate. The one thing we can do is flood the site of a reaction with lots and lots of enzymes. And so we don't need to have a higher temperature. We don't need to have more reactants. We don't need to have smaller particle sizes because we already have smaller particle sizes and low concentrations. We're stuck with that. So what the enzyme does is the enzyme has an area called it has a reactive site that the substrates are what we call the reactants. all an enzyme does is create a location for the reacting material to land on. So if you have low concentrations of reactants, but if you produce lots, if you have a very low concentration of reactants and you are depending on those reactants to just gradually run into each other and bounce into each other, you know, that reaction can take forever to occur. But if you suddenly produce lots and lots of these enzymes, and it has a reactive site there where it can bring the reacting material together in place, then the reaction can go faster. So as we see in the animation here, there's our enzyme in pink. And we have two substrates. It can be more, let's just say two substrates that come together. They fit perfectly into the reactive site on the enzyme. Nothing else will fit there. Enzymes are specific for a wide variety of molecules. So the substrates can land on the enzyme and they'll just bounce in there, but you see you've got millions and millions of enzymes in the space. And so these reactants land on the enzyme, physically brought together, and now they'll react. And when they react, then the enzyme will let go of the product and then go do it again and again and again. Yeah. I've done this actually in class. If we were in a great big empty room and there were two people and there's no furniture in the room, no desk or anything like that, take two people, blindfold them. You don't have to spin them around, that makes it even better, but blindfold them and turn them loose from opposite sides of the room and say, okay, you need to walk around until you encounter the other person and shake hands with them. Okay, so that's what's happening without enzymes. You know, in our bodies, with the small concentrations of reactants that we have and the small particle sizes we have, you're going to wander aimlessly throughout the room trying to randomly encounter the other person and shake their hand. But what if everybody else in the class was an enzyme for those two reactants? And so we put everybody in this big empty space with two reactants. They're trying to walk around blindfolded, trying to find what they're looking for. And now all you have to do is, as an enzyme, you walk up to them and bring them together. And they shake hands. And then you walk away. And then someone else comes up and they shake hands. And someone else comes up and brings them together. And so they're randomly circling around and you just keep bringing them together and bringing them together. That's a whole lot faster and quicker than waiting for them to randomly move around and bump into each other and shake hands. That's what enzymes do. They bring the reactants together simply by being there in large quantities. Enzymes always end in an ASE. You say amylase, catalase, lipase. It's an enzyme. Always has the ASE ending on here. Enzymes usually have two parts. The protein structure of an enzyme is called the APO enzyme, and there is a cofactor or coenzyme attached to that. And that coenzyme is usually where vitamins end up. That's why we take vitamins. Vitamins become a component of an enzyme to make it work better for us, So enzymes are very, very specific. The reactants, we call them substrates. And so the enzymes are substrate specific. Amylase only works on starch. Lipase only works on lipids. Catalase only works on carbohydrates, to break it down. So different enzymes do different things. They're all going to be unique. Amylase that works on starch in our mouth is not going to be able to work on protein in our stomach. It won't fit. The protein molecules don't fit into the amylase. So wide variety of types of proteins. Everyone is substrate specific. And I guess this is probably this is probably a really good place to stop. We'll end up wrapping up chapter 2 on Thursday. So we're going to finally be done with this chapter. But I will go ahead and end this now. And I will lab this week. We're going to be looking at the appendicular skeleton for bones. So expect to look at that today. And I will see some of you in lab then. So I'll get us out of here and I'll see you in lecture on Thursday.